Hello, in this video we will discuss about the flow cytometer. So what that means? Flow cytometer. Flow means the movement and cyto means the cell and meter means the measurement tool. While here is we can say the simple is the FACS, fluorescent activated cell sorting mechanism. So this method is known as the FACS or FACS. So this basically is used for the cell counting. It means the number of cell we can count. On the other hand, the analyze phenotypes. Phenotypes mean the shape of the cell, and genotype, you know about that. The genotype is the inside the gene relating things, but phenotype is the appearance. And health of the cell and cell size you can measure, and cell complexity you can measure. So here is the person which that is, for example, suppose this is the patient and its patient contain a basically different type of cells. White blood cell contain a neutrophil, eosinophil, basophil, macrophage as well as monocyte, and on the other hand the red blood cell contain RBC and platelets, which that is used for the blood clotting. So platelets is not a basically cell, but it is the basically protein. So thrombin protein is. So we will take the sample from the patient, and this sample contain a different type of cells. So this sample is used for the detection of the cell size, cell phenotypes, and cell health and cell size as well as cell complexity and cell counting. So here is the flow cytometer, which that is a simple diagram for understanding the flow cytometer is here, which contain a sheath fluid is here, and that is inside of the inside where we will put the cell is saline solution, which that saline solution will be separate the cells. And hydrodynamic focusing we will discuss. Hydrodynamic focusing means the movement of cell one by one to detect it by a laser beam. So this laser beam will be when put this sample one by one cell. So there are different cells which scatter the light by a striking with a laser beam. So when laser beam will strike to the cells, it diverge. It means the side direction and forward direction on the basis of complexity and cell size. So here is the we put the detector for detecting the light. When the side direction we receive the side direction light dispersion and falling on the detector, while on the forward detection we will receive by the forward detector. So this detector is basically different types which can be measured with the analyzing unit is known as a PC, and this PC can show the graph or something, which that is we can indicate the problem as well as size, complexity, health of the cell. So let's begin to understand how to detect. So here I am adjusting the laser beam on the small capillary of the cells when cell will be moved one by one. So in the forward side the detector, this detector which that is detect the direction of the light, either move more wider or closer. So it means the cell size is directly proportional to the scattering light. It means the cell will be larger. It means the scattering light will be more. It means we can detect. While on the other hand, the smaller cell we can put the laser beam is here, which that is we can again check it to smaller size will scatter less light. So the detector can be detected by the intensity, more intensity due to the more voltage. So the more voltage will create the more intensity, so that PC can analyze and show the complexity and granularity. So we will also understand the complexity and granularity on the basis of scattering of light more. So when complexity of the cell mean the more protein are present on the surface of the cell and granules inside, both things which is responsible for the scattering of more light from the cell. So you can see that On the other hand, here is the graph which that is showing in the figure. Intensity of light is the horizontal while the voltage is the vertical. You can see that when the large cell wider, so the large cell is the vertical is the wider, so the large cell basically produce the wider direction of the light scattering, while the small will produce the less on the basis of time. So here is important thing is that the complex cell is neutrophil, eosinophil and basophil are present in the white blood cell, which contain a granules, and that is known as the granulocytes, and on the surface of their plasma membrane contain a different type of protein, for example receptor and antigen or something. So these on the basis of complexity scatter more light as compared to the simple cell, for example the red blood cell. So here we can detect by this graph to scatter side scatter light is the vertical showing in the figure with the on the basis of nano angle, which that you can see that's 200 to 1000, while on the other hand the horizontal line show that the forward angle scattering light, so the forward and side scattering light. So here is the four boxes can be appear to understand as well. So the number one is here box is show that cell is simple and small. Why? Because it scatter less light on the side scattering light, and forward is also smaller angle, but the wider basically side scattering light is also smaller, so it means the cell is simple. While on the other hand the blue color shows that cell is small and complex. Why? Because side scattering light is more, and on the basis of forward angle light is less, so that means you can say that it is higher number of protein molecule are present on the surface, so we can say that is the complex. So on the other hand, here is the cell is complex and big. Why? Big cell will produce the side scatter light more as well as forward angle light scatter more. So the angle of the forward and side will be more. So it means the cell is complex and big. But on the other hand, the black color shows that cell is small, not big. Here is my mistake. So the cell is small and simple. Why? Because it is small, it scatter less light, and it is simple. Simple mean it is forward light is more, but on the other hand the side scatter light is less. So here that was the 3D histogram to understand the cell structure. So the 2D. So the 2D histogram I am drawing again to understand the white blood cell on the basis of their surface protein, their complexity, and their smaller and larger cells. So here is the horizontal show the forward scattered light, which that is we are detecting the sample of the patient. So here is the side scattered light is the vertical. So show the angle on the basis of scattering of the light from the laser beam to strike in the cell to diverge, and side scatter and forward scattering of the light. So here is I am drawing again the four boxes to understand. Here is the platelets. Platelets is very small and other cell debris will be occur here, which that is very simple. It means it is cell is simple and small. So the smaller particles show in this box. On the other hand, here is you can see that the monocyte, which that is basically is the forward scatter light is more, but the side scattering light is less. So it means important thing, it means it is monocyte and it is smaller. But on the other hand, here box shows that cell is big and complex because scattering of the light side and forward is more wider. So you can see that eosinophil, basophil and neutrophil is the complex, and their surface contain a different type of protein, and inside the many granules which that is scatter the more light side and forward is wider. So that means we can identify that is a complex. But here we will discuss about the next is that was a flow cytometer, which that is used for the mechanism of detection of the cell and their complexity analyze phenotypes, health, cell size, and cell complexity. So here is the FACS, which that is the fluorescence activated cell sorting method. So it's on the basis as here is the sample cell mixture heterogeneous. Heterogeneous means the different type of cell are present in our blood. So these blood contain a basically we are preparing the antibody containing a inflorescence producing fluorophore, which that is attached with the antibody to show the complexity on the basis of producing light. So here is the protein on the surface of the membrane of this different type of cell, which that is differentiate on the basis of their surface protein as an antigen or receptor. So that means here is the antibodies are against interest basically against interest of antigen. So we are preparing to understand that is against the antigen. It means we are understanding that antigen are present in that cell, and we have these antibodies, which that is you can buy from the market, so that we will attach the fluorophore in the lab to detect it. So if these antibodies are against the antigen of that cell, so it means we can bind this antigen with the antibody complex to show that structure. So these antibodies contain a fluorophore, so fluorescent luminescence. So this is scatter more light forward and side. So that means it means we can detect this light by a different direction. So we can use the detect fluorescence by the cytometer to understand. So let's begin to understand. First of all, here is I am drawing the basically sample. First of all, it provides a method for sorting heterogeneous mixture and biological cell. So we can provide a method for sorting heterogeneous. So heterogeneous means a different type of cell mixture. So this cell mixture as a sample will add with the antibodies, and this antibody will be bind on the basis of their specific surface protein. So that means it is also contained with a different type of color. So for example, we are attaching the 4 type of colors: blue, green, yellow, and red. So we can separate that color also, but and when separate this color, we can confirm that antigen or molecules are present in the cell, as well as this is the detection of the molecule. It is not show the size of the molecule, but on the other hand, the cytometer, which that is we had discussed in the previous diagram, which that is show that the size and complexity. But here we can say that is the surface protein, which that is used for the confirmation of the presence of that protein. So here I am setting the laser, and that is the simple diagram to understand. So the basically here is the cell flow from the sample. So it means the movement of cell one by one. So the one by one movement of the sample will move, and the laser beam will be strike and to produce the color, and this color will you can separate. How its separation will be occur? So let's begin to understand easily. So here is the basically detector. So the side scatter detector as well as the forward scatter detector. So the forward scatter detector is basically here. So there are many detector, but here I am drawing just for understanding. So the side scatter detector light and forward scatter detector light. So these you can watch this. So here is basically when fall the cell is downward the one by one drop wise. So it means the basically you can forward scatter light and use. So the side scatter light will be separate in the different side scattered detector, which that is fixed, and here you can see this in this diagram, and dichroic are present, which that is inhibit the forward light, and that means the separation of the light in this mechanism. While on the other hand, important thing is that this detector will be bind or will be attached with the CCD or computer detector, which that is we can analyze it. So this means this important thing is here, that is computer show the color. This color will be show on the basis of graph to understand, right? And that means the color will be shown on the basis of presence or absence of the protein. If for example, if red color is not showing in the PC, so it means the red color relating antibodies, which that is the fluorophore is attached, are not present. After the washing will be washed away, so it means the antibody is not present. So here is our choice of antibodies against the protein, so we can detect. If not bind with the cell, so not color produce that. So that is things. So I hope you make sense. So the forward scatter light is directly proportional to the size, and side scatter light is directly proportional to the complexity. And fluorescence gives protein expression for detection, either protein is present or not. So thanks for watching. Please make sure to subscribe, like and share. Ask the question for answer. So thanks. Bye.